Abundance, vertical distribution, and vegetative preferences of salt marsh snails. I hypothesize that salt marsh snail dispersion is random, with no specific preferences in vertical location and vegetation, and species abundance is equal across the study site. Let's meet the snails. Polygyra cereolis is the common name of the southern flatquail. The shell is shaped like a disc and the foot is yellow. The shell is white with streaks of gray or brown. It has 7 to 9 whorls. It is 3.5 to 4.6 millimeters. It has no gills and a paleo lung to breathe air. The shell top may be flat or pointy. The southern flatquail consumes clover and alfalfa and is native to Florida. Melampus coffea is commonly known as the coffee bean snail. The shell is ovate or teardrop shaped. The coffee bean snail migrates vertically as the tide rises. This allows them to escape predation and water. The snail is brown in color with lighter colored bands. It can be gray, tan, or yellow. The aperture is long and thin. It does not have an operculum, which is the door that closes the snail in. The coffee bean snail lives in intertidal areas along the coast of Florida and the Caribbean. The coffee bean snail will live around red, black, and white mangroves. They don't have gills and they breathe through a hole in their mantle cavity. The coffee bean snail eats detritus and mangrove leaf litter. The coffee bean snail is preyed on by white ibis. Literaria aerata is commonly known as the marsh periwinkle. The marsh periwinkle's preferred habitat is Spartina and Juncus. Fossils suggest that in a post-glacial period, Literaria aerata expanded up to New England. This snail lives in brackish or saltwater. The shell is ovate and conical. It has 5 whorls and a pointed spire. Adults can be 20 to 32 millimeters. Literaria angulifera is known as the mangrove periwinkle. The shell can be bluish-white, orange, yellow, red-brown, gray-brown, or gray-white. The mangrove periwinkle often has stripes or spots and 6 whorls. At max, the mangrove periwinkle can be 3.2 centimeters. The shape is elongate conic, like a long oval with a point. The mangrove periwinkle has 8 to 20 whorls. The mangrove periwinkle has a range that extends from Florida to Brazil. Literaria angulifera is ovoviparous. Their eggs hatch inside their body and are released as larvae. The mangrove periwinkle grazes on algae and fungi. Some snails have a home site which benefits them more than foraging in the substrate and protects them from predators. The shade of the mangrove trees keeps Literaria angulifera from desiccation. So for my message, I walked around the 312 salt marsh, excluding areas that were too muddy to sample, and I looked in 3 main places. Shaded Spartina, sort of this line of mangrove trees, and open sun Spartina. And I found that the species ended up choosing locations that were specific with lots of the vegetation that they prefer. I sampled 2,454 snails. So, coffee bean snail here, and we're going to talk about Melampus coffea. So, of the coffee bean snails sampled, the lower third was preferred 46% of the time. Melampus coffea's preferred vegetation type is Spartina. Their second choice is Marima, which is actually called saltwort. It's a succulent plant. Both days the coffee bean snails were spotted were on high tide days at 80 and 81 degrees. So we know that coffee bean snails prefer the lower third and they prefer Spartina. Next, we're going to look at Literaria angulifera. Literaria angulifera preferred the upper third of black mangrove trees. Black mangrove leaves were more preferred than black mangrove stems. This may be because leaves are an easier food source with access to green organic matter. Mangrove periwinkles preferred mangroves 47% of the time. but they preferred Spartina 45.6% of the time. That shows that the mangrove periwinkles do not have as much vegetation preference as Literaria aerata. Polygyrus cereolus preferred the lower third 78% of the time. They preferred water 64% of the time, more than any other marsh snail, so they want to be in the lower third in the water. Their second choice was Spartina at 21%. So next we're going to look at the chi-squared test. So we looked at, Dr. Grant and I looked at vertical type of plant and type of snail. We found that the p-value was less than 0.5. That shows an association between vertical location and type of snail. That verifies what we saw in the bar graph. That means that snails prefer different heights. Literaria angulifera prefers black mangroves, and Melampus coffee. The p-value is less than 0.5. That indicates an association between vertical location and type of snail. This verifies the bar graph evidence. Dr. Grant and I ran a chi-squared test between type of plant and type of snail. We found that the p-value is less than 0.5. That shows an association between type of plant and type of snail. These graphs show Literaria angulifera prefers black mangroves, and Melampus coffea and Literaria aerata prefer Spartina. This was a really cool study that I read about that was about top-down control. So it said that radulating creates wounds on live Spartina. These wounds kill more biomass than is consumed by a snail and slows plant growth. Periwinkles only eat less than 37% of Spartina, but the whole stem is damaged. Snails have a top-down control on Spartina biomass production. They keep the species in check. It is possible snails cause a microbial infection in Spartina. Snails graze on living Spartina even when dead Spartina is available. Snails create and maintain wounds with their radulas and consume dying tissues instead of green or dead tissues. Radulations per stem nearly doubled when the marsh was fertilized. So in this study, they had a snail removal trial, a resident snail trial, and a resident snail plus another group of snails trial. And they did all these trials again, including nitrogen, and they found that snails eat more vigorously when there's more nitrogen. So when the Spartina is growing even more, snails eat even more to keep this top-down control on Spartina plants. Literaria angulifera had the highest abundance at 51. Literaria aerata had the second highest abundance of 36. Melampus coffea had a species abundance of 12. And Polygyrus cereolus had an abundance of only 6. The overall preferred vegetation was Spartina. This is because Literaria angulifera and Literaria aerata are the two largest groups of snails, and they're both found on Spartina. The second largest preferred vegetation was black mangrove leaf. That is because it is the top choice of Literaria angulifera and the most abundant species in my sample. So here you can see all salt marsh snails and the percentages of them in my total sample time. Next, we're going to look at vegetation preferences. That there was only one snail that was on a mangrove seed. Put a picture of him here. In general, snails stay pretty consistent. Spartina is most preferred, black mangrove is the second, and that's just because Literaria aerata and Literaria angulifera prefer those places the most. I found that the upper and middle thirds are the most preferred vertical location. The most abundant species, Literaria angulifera, prefers the upper third. The second most abundant, Literaria aerata, prefers the upper third only 5% more than the middle third. Because these two snails dominate in preference for upper and middle third and have high abundance, their general vertical location preferences outshine other species and other locations. I was wondering what they eat, so I looked it up, and it said that snails sometimes eat algae and detritus that builds up in the pores of rock. Literaria aerata climb and forage at low tide and go into the sediment. When foraging in the sediment, snails tend to stay about a centimeter away from the Spartina stem. In high tide, they climb to the upper portions of the cordgrass. Juveniles stay closer to the water than adults do to forage. This is because the base of the plant is not associated with temperature and desiccation stress. So, I did a mini study to see if I saw more juveniles in the lower third. I saw a trend for juveniles in the lower third. In warm weather, snails forage in the substrate. The 29th was my warmest sampling day, but I didn't see an increase in marsh snails in the lower third. This is a chi-squared test between vertical location and tide. Since the p-value is greater than 0.5, the null is true. There is no association between tide and vertical location of Literaria angulifera. The tide does not affect Literaria angulifera's vertical location. This is different than the graph of my observation, but that's what the statistical test shows. This graph is about seeing if there were different vertical location preferences with the tide. In my study, on high tide days, mangrove periwinkles preferred the upper third. On low tide days, mangrove periwinkles preferred the middle third. Spartina periwinkles preferred the upper third despite the tide. I also saw that it's possible climbing is not associated with avoiding the tides, but associated with. Next is a chi-squared test of vertical location and tide. I found that the p-value was less than 0.5. That shows an association between the tide and location of Literaria aorata. But after that, I found that rainy sampling days, the 22nd and the 27th, there were more snail pairs. I wrote this down and asked Dr. McGinley, and he said it could maybe be breeding behavior to see if surface preference was less specific on rainy or low tide days. This keeps Spartina in check, but overgrazing from the addition of Literaria angulifera may cause an even greater stress on Spartina health. Manatee route. The St. Augustine water treatment is in the natural manatee route. St. Augustine water treatment is a stop, and manatees feed on marsh grasses and algae in the 312 salt marsh. Climate change can have a huge overarching ecological effect. To protect the manatees, reducing carbon emission can help save Spartina, snails, and manatees. Polygyrus areolus and Melampus coffea were both spotted on low tide days. Literaria angulifera did not vertically migrate with the tide, but Literaria aorata did. That shows snails have preferences. So we know that periwinkle snails have preferences. We know that they have unique abundances, and they prefer different vegetations, and they prefer different things based on the tides. A big thank you to all the people who helped me. These are my citations. Thanks for watching.